Behavioral Neuroscience, Chapter 14, Segment 1. Biological rhythms. Biological rhythms range in length from minutes to seconds or from months to years. Many animals show daily rhythms and activity. Most functions of any living system display a rhythm of approximately 24 hours. They are called circadian rhythms because they last about a day. Humans and most other primates are diurnal, active during the day, and most rodents are nocturnal, which means they are active during the dark periods. Hamsters are studied on a running wheel, which is connected to a computer that recognizes each revolution of the wheel. Circadian rhythms are generated by an endogenous clock. When a hamster is placed in a dimly lit room, it continues to show a rhythm in wheel running, which suggests that they have an internal clock. The hamster is free running, maintaining its own cycle in the absence of external cues about time of day. Animals have natural rhythms. A period is the time between two similar points of successive cycles, such as sunset and sunrise. Since the free-running period does not match the period of Earth's rotation, and because it differs slightly among individual hamsters in the same room, the period cannot simply be reflecting an external cue. A phase shift is a shift in the activity of biological rhythm, typically provided by a synchronizing environmental stimulus. Entrainment is the process of synchronizing a biological rhythm to an environmental stimulus. Zeitberger is the stimulus that entrains circadian rhythms. An example of that would be light. Circadian rhythms allow animals to anticipate changes in the environment. The major value of circadian rhythms is that they synchronize behavior and body states to change in the environment. The endogenous clock enables animals to anticipate an event and to begin psychological and behavioral preparations before that event. The hypothalamus houses the circadian clock. A tiny subregion of the hypothalamus, called the suprachiasmatic nucleus, the SCN, located above the optic chiasm, serves as the biological clock. The lesions confined in the SCN interfere with circadian rhythms of drinking and locomotor behavior and hormone secretion. Transplants prove that the SCN produces the circadian rhythm. Ralph and Menker found that a male hamster that exhibited an unusually short free-running activity rhythm in constant conditions. Half of his offspring had shorter circadian rhythms, so the researchers concluded that he had a genetic mutation affecting his indigenous clock. The mutation was named tau. Animals with two copies of the mutant gene tau have a period of 20 hours instead of the normal 24. The animals entered the normal 24-hour light-dark period. The abnormal rhythm was only revealed in the constant conditions. Evidence that the endogenous period is produced within the SCN was provided by transplant experiments. Non-mutant hamsters with lesions of the SCN were placed in constant conditions with their circadian rhythms were abolished. The hamsters received transplants of SCN taken from fetal hamsters with two copies of the mutant tau gene. Hamsters then showed free-running activity rhythm again, but the new rhythm matched that of the donor, which is 20 hours. In mammals, light information from the eyes reaches the SCN directly. In some animals, the pineal gland of some amphibians is sensitive to light and helps entrain circadian rhythms to light. Certain retinal ganglion cells send their axons along the retinohypothalamic pathway, splitting off at the optic chiasm to synapse directly with the SCN. These pathways carry info about the light to the hypothalamus to entrain behavior. Most cells do not rely on photoreceptors, rods and cones, to learn about light. Rather, the cells contain a specific pigment called melanospin that makes it sensitive to light. The most sensitive to light frequencies occur in the blue range. Circadian rhythms have been genetically dissected in flies and mice. The fruit fly, Drosophila melanogaster, provides diurnal circadian rhythms in activity. Flies with a mutation that disabled the gene called period PER were arrhythmic when transferred to constant dim light, indicating that their internal clocks weren't running. Subtle mutations of the gene caused the animals to have free-running period that was longer or shorter than normal. The work paved way for understanding the molecular basis of circadian clocks in mammals. Cells in the SCN make two proteins, clock and cycle. They bind together to form a dimer, a pair of molecules attached to each other. It then binds to the cell's DNA to promote the transcription of PER and another gene called cytochrome Cry. The resulting PER and Cry proteins then bind together to regulatory proteins. The PER and Cry protein complex enters the nucleus to inhibit the transition between PER and Cry. It's a 24-hour cycle that drives the 24-hour period of the SCN cells. Each SCN neuron uses this mechanism to keep time approximately, and then all the neurons communicate with each other through the electrical synapses. They synchronize their activity to provide a very constant period of about 24 hours, which then drives circadian processes throughout the body. In fruit flies, light passes through the fly's body into the brain cells to degrade the Cry protein and synchronize the molecular clock. Humans use the retinohypothalamic tract to get light information to the SCN. The retinal ganglion cells containing melasmin detect light and release the neurotransmitter glutamate into the SCN. Glutamate triggers a chain of events in the SCN cells that promotes the production of the PER protein. When the animal's photoperiod is shifted, the light-mediated boosting of PER production cells shift the phase of the molecular clock and therefore the animal's behavior. Some biological rhythms are longer or shorter than a day. Infradian is the rhythmic biological event whose period is longer than that of the circadian rhythm that is longer than 24 hours. A familiar infradian rhythm is the 28-day human menstrual cycle. Some of the rhythms are driven by exogenous factors such as food availability and temperature. Seasonal animals in isolation show free-running annual rhythms of a period not quite equal to 365 days. There seems to be an indigenous circannual clock, which is occurring on an annual basis. The circannual cycles do not arise from the circadian clock. They seem to involve a mechanism that is separate from the SCN. Light cycles can entrain the circannual rhythm by affecting how long the nightly bout of melatonin secretions lasts. Ultradian is a rhythmic biological event whose period is shorter than that of a circadian rhythm, usually from several minutes to several hours long. It is seen in behaviors as bouts of activity, feeding, and hormone release. It may be superimposed on circadian rhythms. Rhythms and performance on various boring tasks reflect fluctuations in alertness, and rhythms of about 90 minutes imposed on the circadian sleep-wake cycle, which will be talked about later in this chapter.